Hey folks, today we're going to explore the wonderful world of proteins. And if you have seen one of the movies based on Lewis Carroll's book Alice in Wonderland or Alice with the Looking Glass, or maybe even read one of his books, his whole idea was about what if you take somebody and put them in a world where everything is completely different. And I want you kind of to think a little bit that way because proteins, as molecules, as all molecules do, they exist in a different world at a different scale than we do. So sometimes the rules don't apply and it can be a little weird. So let's explore this wonderful world of proteins. Okay? All right. Understanding how proteins are built, which is their structure, is a little bit like going down a rabbit hole. Okay? It's a whole new strange world. It's a world about collisions and attractions and repulsions and things breaking and things reforming, things mixing. But what makes this world understandable is we can approach it from the idea of a three-dimensional space or a three-dimensional structure. And one of the most important features of proteins is their three-dimensional shape. So let's explore that. But before we do that, it's important that we review what we already know or have already talked about regarding macromolecules. Okay? Now remember, proteins, like most other macromolecules, are what we call polymers, which means they are built from smaller subunits called monomers. And in the case of proteins, the monomers are called amino acids. Okay? There are about 20 different types of amino acids used by natural systems to build proteins. Now, if you want to make some artificial or some synthetic amino acids, scientists can certainly do that. But in terms of living cells or living systems, we only have to really work with 20 different amino acids. Each amino acid is built exactly the same with some exceptions. It has a nitrogen hooked to a carbon hooked to a carbon in this NCC type backbone. And at one end, at the nitrogen end, there is a functional group called an amine group, which is where amino comes from. And at the other end, on the terminal carbon, we have a carboxyl group, which can dissociate a hydrogen ion and make it acidic. So that's where the acid part of amino acid comes from. Now, in the very middle, in the center, we have what's called the alpha carbon. That is the carbon where what's attached to it can change. And that's what makes amino acids so variable. So let's go and continue talking about this. Each amino acid can have these different attachments, usually at the alpha carbon, and we can call these side chains. And those are where the slightly different properties of amino acids comes from. That's what makes them acidic. Some amino acids are even basic. Some are polar, some are nonpolar. That's all more information. Remember, I told you this is like a rabbit hole. But for right now, we're just going to worry about the fact that amino acids can vary with what's attached to the alpha carbon. Now, proteins get their three-dimensional shape one step at a time. And there are 3 steps for all proteins and a 4th step for some proteins. Now, the really cool proteins have that 4th step, and I'm going to show you some examples on future slides. So let's break it down and take it step by step. The first step, what we call the primary structure, is going to be based on the sequence of amino acids that are hooked together. Now, amino acids are joined to each other with a special type of covalent bond called a peptide bond. Peptide bonds hold amino acids together. And this happens during, you guessed it, dehydration synthesis reactions, because those are the reactions that build polymers out of monomers in living systems. Remember, a dehydration synthesis reaction hooks part A to part B by squeezing out or dehydrating, squeezing out a water molecule, right? Ribosomes and another type of RNA called tRNA or transfer RNA, they work together as a team to assemble amino acids in exactly the right sequence or order for every protein. And that sequence of amino acids is determined ultimately by the DNA molecule. We have a whole unit of study about that idea, but for right now, I want you guys to know that the primary structure of amino acids is determined by the sequence of amino acids, and that sequence has been laid out ultimately by a DNA molecule. All right. So what we call this sequence of amino acids is the primary structure. Now you can write one with a little circle or a little dash after it, which is shorthand for primary. All right. Now, visually, what's happening here is kind of like putting beads on a string. So in this picture, each of the different colored beads represents an amino acid, and the little dashes between them, kind of like the string, those are the peptide bonds. So we can see, to hook together this string of amino acids, we need some peptide bonds to be formed. And I am not going to count all these beads, but whatever number of beads you have, whatever that number is, n minus 1 is going to be the number of peptide bonds that you need to hold them all together into a line, basically. Now, this chain of amino acids can also be called a polypeptide because poly means many and peptide refers to the peptide bond. So we can call this primary structure of a protein a polypeptide. Now, number 2, what happens to this polypeptide? Well, it's going to start taking on a three-dimensional shape, and it's going to do this in 2 or possibly 3 more steps. So the second step is we're going to twist and fold this polypeptide into spirals. They look like springs. The technical name for them are alpha helices. Or we're going to fold it like you would fold a piece of paper into like a fan shape, into a pleat. And these pleated regions are called beta pleated sheets. So some combination of these 2 shapes is what happens next. I'm going to show you a picture, so don't panic. Okay, so each section of amino acids in the chain are hydrogen bonding with water molecules, which are always in the environment, and other hydrogens and oxygens in other amino acids to cause this to happen. Okay, so what I'm trying to say here is if you put a polypeptide, just a chain of amino acids, in water, it's not going to stay straight. It's going to coil and pleat itself up into this secondary structure. It just happens automatically. It's physics. Okay, now conditions in the environment around the polypeptide are going to have a huge influence on how it changes its shape. And there are 3 in particular, and one in particular that's probably the most important: temperature. So pH, temperature, and salt concentration all play a role in how this secondary structure forms. Temperature and pH are probably the 2 most important ones. If you only want to know one of them, if you only feel like you only want to study one, no temperature. All right. If it's too hot or too cold, this doesn't happen properly. Okay, so this is what we call the secondary, or 2 with a little circle or dash after it, the secondary structure of a protein. And let me show you what we're talking about here. So this is a pretty good diagram. I think it's really close to the one that's in the textbooks you would have this year if we were in school. We have a polypeptide, which is what's drawn right here. Okay, and we have different parts of it that have taken on different shapes. So if you look at this part right here, that's what we call, let me circle it so you guys can read it. That's called the beta pleated sheet region. And what that looks like at the molecular level, structurally, is it's like a piece of paper that you folded into like a fan shape. And over here, we have a close-up of what the spiral region looks like, which is called the alpha helix. So what I'm hoping you can see is this polypeptide is going to have different sections that are going to fold in different ways for the secondary structure. It can either pleat itself or it can coil itself into an alpha helix. Now, I want you guys to notice the little dotted lines. Okay, these dotted lines represent hydrogen bonding. So hydrogen bonding is going to be stabilizing the beta pleated sheet. So in this diagram, I am counting 1, 2, 3, 4, 5, 6, 7, 8 different hydrogen bond interactions here, stabilizing that pleat. And the same thing is happening in the alpha helix. Hydrogen bonds are forming to stabilize it into this kind of coiled up spring shape. And in this diagram, I see they're only showing 3 hydrogen bonds. So pretty straightforward. Just remember, the secondary structure is happening primarily because of hydrogen bonding. Okay? Okay, now let's move on to the 3rd level of structure in proteins. Okay? This this depleted sheets and the alpha helices are going to twist and fold themselves again, forming a really complicated three-dimensional structure. So there, we did it. We got the 3D, okay, the third level of structure. And the conformation or shape that this three-dimensional protein takes is very sensitive to environmental conditions. Remember the pH, temperature, salt concentrations. Okay, this three-dimensional shape can be really long, like a filament, like a hair maybe, or it could be clumpy and blobby, what we call globular, or anything in between, or any combination of these shapes. It's almost infinite, the number of shapes that these proteins can take. The three-dimensional shapes for the third level of structure are stabilized by something called disulfide bridges. Now, disulfide bridge sounds complicated, and sometimes students panic when they hit stuff. Oh no, that's chemistry. But it's actually very simple. So I'm going to show you guys in a picture exactly what a disulfide bridge is. And a disulfide bridge, I think it's a brilliant word because the bridge tells you what's happening. You basically have a connection between two places that are otherwise not connected. So let's look at what a disulfide bridge is. And I'm hoping you notice that sulfide refers to the element sulfur. Okay, so we've got to bring sulfur to the party. So this three-dimensional structure of the polypeptide can be denatured, and the denaturization is just a fancy way of saying changed. The three-dimensional structure can be changed by altering those factors in the environment that affect three-dimensional shape. Things like temperature, pH, salinity, things like that. All right. The three-dimensional structure is also called the tertiary structure. Tertiary refers to the number 3. Just in case you read or hear that, I don't want you guys to panic. Tertiary just means 3. All right. Now, here's a pretty good picture of a kind of salmon-colored, pleated and spiraled polypeptide, showing you all the different things that can hold it into a three-dimensional shape. And the only one I'm really worried about right here for AP Biology is the disulfide. Oh, I did a bad job circling that. The disulfide bridge, which is right here. So I'm hoping you can see two sulfhydryl groups on two different amino acids become close enough to form a bond between them, a covalent bond, which gets a fancy name. It's called a disulfide bridge. So there's one there, and there's another one right here. And these stabilize the three-dimensional shape of the protein, along with other things that I'm not really going to worry about this year. All right, so, I already did it. So here we go. There are the two disulfide bridges circled in blue for you. All right. Now, there is an amino acid called cysteine, which has a sulfur attached to it. And this sulfhydryl group is right here. Okay. So that sulfhydryl group is connected to that alpha carbon in the middle. So I'm hoping you guys can see that. So this sulfhydryl group gives this amino acid the ability to form half of a disulfide bridge. So if we bring two of them close enough together, they can form that little connection that we've been talking about. So I'm hoping you guys can figure that out. Now, I think a real world example is really appropriate for right now. Everybody has hair, or at least somewhere on your body you have hair, probably on your head. Probably the biggest part of human culture, I think, is just imagine across the world what people do with their hairstyles through time and through space. But hair on humans is all made from the same stuff. It's a protein called keratin. And keratin is an amazingly resilient protein. It's tough and flexible and waterproof. It is amazing stuff. It resists bacterial digestion. It resists rotting. It resists ultraviolet radiation. It resists acids. It's tough stuff. And the toughness of keratin is coming from all the disulfide bridges that it has built into it. So if a protein is tough, then it must have a lot of disulfide bridges. And if it has disulfide bridges, it must have a lot of cysteine in it. So it must have a high proportion of that amino acid in its primary structure. So I'm hoping you guys can see how structure relates to function when you drill down to really fine levels of discussion here. Now, I didn't tell you guys the whole truth. That keratin molecule actually has 4 levels of structure. Now, I didn't lie to you. I just didn't tell you the whole truth at first, but I want to straighten that out right now. Some proteins have a fourth level of structure, all right? And some polypeptides combine with other polypeptides to make a higher order structure. And what that means, simply stated, is some proteins are actually made up of multiple primary structures that become tangled up with each other. And I'm going to show you some pictures of a couple in just a second. All right, this is called the quaternary or fourth degree of structure in proteins. So I don't want you guys to freak out over the word quaternary. That's where that's coming from. It means four. I think it looks a little bit like the word quarter. That's how I've remembered it. All right, now keratin actually has a quaternary structure, and it's made up of multiple primary structures or polypeptides that are disulfide bridged together to make it really tough and really strong as a hair. Keratin is also the main ingredient in fish scales and toenails and fingernails and rhinoceros horn and feathers. Those are all made of keratin. Biology, or vertebrates at least, have really latched onto this protein molecule as a good building material for all sorts of different body parts, primarily for protection or things that are at least exposed to the environment. So I need to stop. Don't want to go too far down that rabbit hole, but biology is cool, and keratin is a really cool molecule. Quaternary structure. Let's look at a different one. Keratin's pretty cool, but hemoglobin is fantastic. Look at this picture. That is just the coolest thing. I mean, you might want to print that and frame it and hang it on your wall. This image was drawn by a computer that was programmed with the information it needed to properly fold a protein. And so, based on decades and decades and decades of work, scientists have built computer programs that if you plug in the primary structure, basically the genetic code, it will spit out the shape of that protein in whatever pH and temperature you want to put in as parameters, which is really, really cool. These are called protein folding programs, and you need a supercomputer to do them because the variable number is so high. But this is a hemoglobin protein, and it's actually made up of 4 different polypeptides that are all entangled with each other, but not in a random way, very specifically combined. And they're color-coded here, so there are 2 red ones and 2 blue ones. So altogether, they kind of make a donut shape with kind of opening in the middle. And it's this 3-dimensional shape that allows hemoglobin to do what it does. And some of you may associate hemoglobin with red blood cells because it's the protein that actually picks up and releases oxygen depending on where that blood cell happens to be at any moment in time. So hemoglobin can pick up and release oxygen atoms at different concentrations in different ways, depending on whether it's in your lungs or in your muscles. Okay, we'll talk more about that later. pH controls it, so I'll just drop that right there and leave it for right now. But pH in the environment is what's controlling how this molecule behaves. And I can't go without telling you guys, if you look really closely, you'll see these other structures here, which are kind of in a green. Those are the heme groups, and they contain the element iron. They have iron atoms in them, and it's the iron that makes this whole thing work. So that's why if you've ever, got a busted lip or tasted blood, it has a metallic taste. It's coming from the iron in the heme groups of your hemoglobin proteins. You see, biology is so cool. All right, let's wrap this up. So, some questions. I put this slide in because at the end of this video cast, remember, you want to be able to talk and write about this and answer questions about it. So let's look at some possible questions. First thing, what determines the primary structure of a protein? Do you remember what determines the primary structure of a protein? Can a polypeptide achieve its secondary structure in a really dry environment or a dehydrating environment? What's denaturization? What does that word mean? What is protein folding? What are people talking about or writing computer programs about when they talk about folding proteins? What environmental conditions can interfere with how a protein folds? Okay, I gave you guys 3 of them. If a protein is folded incorrectly, what effect will that have on its function, if any? Okay, because remember, structure or shape is intimately connected to function at every level in biology. It's remarkable how important that theme is. All right, so we come to the end here. Thank you, Lewis Carroll, for letting me latch on to your creativity here. I'm going to close here with one of my favorite quotes from Alice Through the Looking Glass by Lewis Carroll. I think it's a great thing to think about sometimes, and my favorite character from Alice in Wonderland, right there up in the picture. But thank you guys, and I'm going to stop there.